Hi everyone, this is section 22.4, the genetic basis of cancer. In this particular one, we're going to talk about the different ways that oncogenes or the loss of tumor suppressor genes can lead to cancer. Often we see these working in conjunction, meaning that an oncogene is basically turned on and we have loss of tumor suppressor genes as well. We're going to talk about the key characteristics of cancer and then compare and contrast those oncogenes and tumor suppressor genes. This particular part of the chapter is just an overview. We'll get into more depth later on in this chapter. So just knowing what cancer is a disease that is characterized by uncontrolled cell division. So generally we think of cancer as cell division that does not stop when it should, and it's a genetic disease at the cellular level. Human cancers can be classified by the type of cell that's become cancerous, and so we often name them by the type of cell, whether it's liver cancer or breast cancer. It's the actual type of human cells that have gone through this uncontrolled cell division. There have been more than 100 different kinds that have been identified so far. So when we think about the characteristics of cancer, we want to think about some key things. So the first thing is to think that most cancers originate from a single cell, right? So when I talk about uncontrolled cell division, it starts out with one cell. And that growth is what we call clonal in origin, in that that single cell then is cloned and replicated. We also want to think about cancer as a multi-step process, that it starts out as what we would call a benign growth, meaning that it's not invasive, it's contained in the area that it is. And then we can have additional genetic changes accumulate over many years. As these genetic changes accumulate over many years, we can see that cancer often will become what is called staged, meaning that we start out at this benign level, but over time, as more genetic changes happen, it begins to cause more and more problems. And so the stages that we would see as benign, we would move into what we consider malignant, which means that it's invasive. It starts to invade surrounding tissue. So that clonal origin begins to move into surrounding tissue, not just that one area. And then lastly, that third stage that we would see is what we call metastatic, and this means that it actually moves to a completely different sites in the body. Okay. So I have a quick kind of graph of this. So we see up at the top, we have our initial tumor cell. You can see it's here. It begins to go through benign growth, meaning that that cell replicates itself many, many, many times, but it's still staying within that same tissue. What we can see, though, is that we get invasion of surrounding tissue and metastasis. So invasion of that tissue is the malignancy, but when we get metastasis, it means that some of these cells break off and enter the bloodstream and move to another part of the body. So just some statistics to keep in mind when we think about cancer. There's over 1 million Americans are diagnosed with cancer each year, and almost half of them will die from the disease. About 5 to 10% of the cancers are due to an inherited predisposition, meaning that there's some sort of inherited genetic component that means that those people are more predisposed to get cancer. But that tells you then that 90 to 95% of this is not something that is inherited predisposition. A small subset are the result of spontaneous mutations in viruses. So spontaneous mutations, we want to think about replication of DNA, that there's a spontaneous mutation. Or virus, often we see these virus infections that can actually be what we call oncogenic, that they can actually cause cancer. Most, though, up to at least 80% of the cancers are related to exposure to mutagens, something in our environment. And these actually alter the structure and expression of genes. So any environmental agent that causes cancer is called a carcinogen. So the two genes that we're going to be looking at more in this semester are oncogenes and tumor suppressor genes. And these are genes that are involved in cancer. So the ones that are considered oncogenes are mutant genes that actually are overexpressed, and they basically are telling the cell to go through uncontrolled division. They contribute to that cancer growth. So oncogenes are the ones that basically tell the cell continue to divide. The other type of genes that are involved in cancer are what we call tumor suppressor genes. And these genes work to prevent cancer, meaning that if they see uncontrolled growth, they tell it to stop. what we can often see is that we could have an oncogene that is overexpressing, meaning that we're getting uncontrolled growth, and then we get loss of function in a tumor suppression gene that allows that cancer growth to occur, meaning that that tumor suppressor gene isn't being expressed, and so we no longer have that suppression of cancer. And often what we see is an accumulation of these types of genes over time. So we may have a mutant gene that's overexpressed from very early on in life, but our tumor suppressor genes have taken care of it. As we age or we're in contact with carcinogens, perhaps we get that loss of function in our tumor suppressor that now this oncogene can go ahead and be unchecked and cause cancerous growth. So that's the end of this particular section. We'll be moving then into the oncogenes in section 22.5. Please let me know if you have any questions. Thanks.